Hello, welcome to all in the next presentation on the topic enzymes. In this topic, we will discuss about mechanism of action of an enzyme. To study mechanism of enzyme action, one should have to remember a basic equation of a reaction, and that equation is enzyme plus substrate. It forms ES complex, that means enzyme substrate complex, and that enzyme substrate complex later on get converted or forms a product, and enzyme is again liberated. That is E plus S, it gives or it forms ES complex, and ES complex forms product, and again enzyme is liberated. Now you are observing here two structures on your left side of the screen, and these two structures, among those one is of yellow to orange color and other is green color. The yellow color structure represents the structure of an enzyme. You can observe that the enzyme has active site, and the green color substructure is that of substrate. Now, at prior state, enzyme and substrate, they are in separated state. Later on, in the diagram, we observe that there is an association in between enzyme and substrate, and they form ES complex, that is enzyme substrate complex. Then the question arises that which different things leads to the formation of ES complex. Now we will think about the formation of ES complex and the parameters or the factors which are responsible for the formation of ES complex. And the answer is catalysis. Now there are various types of catalysis. First one, acid-base catalysis. Second, covalent catalysis, and third, entropy effect or proximity effect. Before that, we have to study the structure of an enzyme. The enzyme structure is represented by means of green color. We observe the U-shaped structure showing clip where the substrate get fitted. That clip has active site. Remember one thing, that out of total surface area of the enzyme, active site possesses about 5% of the total surface area of the enzyme. That means active site possesses only 5%. Then typical structure of enzyme, it has active site in which substrate get fitted. Now how the substrate get fitted into the active site? For that purpose, we have to study first one, acid-base catalysis. Now we know that enzymes are proteinaceous in nature. That means in their structure they possess proteins, and proteins in their structure, they are made up of amino acids. And we know that 20 naturally occurring amino acids, they are classified on the basis of presence of acidic groups or basic groups present in their structure, as well as positively charged amino acids, negatively charged amino acids on the basis of presence of specific charge in their structure. That's why at the active site, where active site of the enzyme, the amino acids either possess acidic group or basic group in their structure, and those amino acids leads to formation of acid-base catalysis. Now the on the structure of substrate, specific groups are present, and those specific groups tend to get united or tend to get reacted with the acidic or basic group of the amino acid present in the active site of the enzyme. So acid-base catalysis tend to formation of enzyme substrate complex. Next one, covalent analysis. Just I have mentioned that amino acid are positively charged as well as negatively charged, and such amino acids present at the active site of an enzyme, they tend to form an association with the substrate. That's why, due to such charges, enzyme substrate association takes place in between active site, or rather to say enzyme and substrate. Next one, proximity or entropy effect. This relates with the number of either substrate or enzyme molecule, and it can be stated that as the number of substrate molecule increases or enzyme molecule increases, there are more chances of formation of enzyme substrate complex. This relates with proximity or entropy effect. Next one, we will actual study the mechanism of enzyme action. Before that, we have to study the meaning of activation energy and transition state. Now, this is graph which is plotted against progress or rate of reaction with potential energy of the substrate. You are observing, or we are observing, two lines. First one is red colored and another is pink color. Both the lines tend, or both the lines refers to the journey of substrate up to the formation of product. At initial state, substrate is there, while at final state, product is there. That means substrate is converted into the product. And these two lines, these are nothing but the journey of conversion of substrate into the product. Now we will discuss about, or we will concentrate on red line. Here in red line, substrate, as the substrate is converted into product, the potential energy is increased. Why there is increase in the potential energy of the substrate? Because this journey, remember the journey related with the red line, this journey or this reaction takes place in the absence of enzyme. Now we will concentrate on pink line. Same reaction is there, substrate is converted into the product, and only difference is that here there is action of enzyme over the substrate. Now, by means of enzymatic activity, activation energy is lowered. We observe the difference in between activation energy in both the lines, or both the reaction with the enzymes and without enzyme. In the absence of enzyme, activation energy of the substrate is increased, or activation energy is increased, while in the presence of enzyme, activation energy is lowered. Now, another one word is there, that is transition state. Enzyme favors the formation of transition state. Transition state is nothing but, this is a state in the reaction where, or from that point onwards, substrate is definitely going to be converted into product. For example, concentrate on other image present on right side. There is a man, and the destination, or he want to reach his destination. There are two ways. First one, he has to climb the hill and then he will reach the destination. During this journey, he will require more energy. But if there is presence of a tunnel through that hill, he will require less energy. The same role has been carried by the enzyme with respect to tunnel. It lowers the activation energy in the reaction. Next one, we will study mechanism of enzyme action. There are different theories. We have mentioned here four different theories. Those are enzyme substrate relationship theory. Second, Fisher template model or lock and key model. Third one, induced fit model or Coastland theory. And fourth one, substrate strain theory. We will first discuss about enzyme substrate relationship theory. As earlier mentioned, I have stated that you should have to remember one equation, and that is nothing but the E plus S. It forms ES complex, and ES complex turn into product and enzyme. That is, S plus E, or E plus S gives ES complex. It forms P plus E. Enzyme substrate relationship theory has been put forth by Leonor Michaelis and Maud Maintain. This is simple model which relates with the association of enzyme and substrate. Why enzyme and substrate? They are associated, that already we have studied in previous slide, and that is nothing but the acid base catalysis, positively charged, negatively charged catalysis, that already we have discussed. Next one, second theory is Fisher template model or lock and key model. This was proposed by Emil Fisher in 1894. The basic principle involved in this mechanism or in this model is that as specific lock can be opened by a specific key, in the same sense specific enzyme acts on particular substrate. For example, enzyme sucrase will act only on sucrose. For example, enzyme lactase will act on lactose, and so on. This model relates with physical contact of physical contact in between enzyme and substrate. There is rigidity in shape of the active site of an enzyme. For that purpose, we have to concentrate on the diagram which is present on right side. Here we are observing the green color structure, that is of enzyme, and in front of that enzyme there are three structures. Among those, two have been represented by red color and one is with blue color. These three structures are of substrates. Now look again back to enzyme structure. Here there are two constrictions. These two constrictions relates with the active site of that particular enzymes. Now, among these three substrates, the blue color substrate, it will get fitted properly into the active site of this enzyme. That's why blue color substrate, it will form ES complex with the enzyme, and then then only product is formed and enzyme is again liberated. The two substrates which have been designated or represented by red color, they will not get fitted into the enzyme, as Emil Fischer has stated that there is rigidity in the structure of enzyme active site. Enzyme active site doesn't change its shape. It is rigid. That's why only blue colored substrate will form ES complex. This theory has been supported by various facts. Among those, the various evidences tell that during the physical contact, enzyme substrate complex is formed because each enzyme has a surface upon which substrate is accurately fitted. Second evidence is that in competitive inhibition of enzyme, the active site of enzyme is blocked by another substance which has near about same structure as that of substrate, and it results loss of enzyme activity. These two evidences proves the uh, advantage of lock and key model. But this lock and key model failed to follow the Michaelis and maintenance constant. That's why it is not widely accepted. Next theory, that is induced fit model or Koshland theory. This was proposed by Daniel Koshland in 1958. This relates with flexibility and modification in the shape of active site as per active site of the enzyme as per the substrate. For example, please concentrate on the diagram on left side. Here the substrate is represented by green color structure while enzyme is yellow color structure. Now concentrate on other side. ES complex has been formed. Again, now concentrate on the structure of substrate. There is no change in the structure of substrate at prior as well as later state, but the enzyme active sites, they modifies themselves as per the structure of substrate. This is the main principle which is involved in induced fit model. Induced fit model states that there is flexibility in the active site of enzyme. Enzymatic active site change themselves as per the structure of substrate. Thus, this model suggests a kind of hand and glove relationship between an enzyme and substrate. Now, explanatory examples. We will discuss only one. Concentrate on balloon and smartphone. Here, balloon is playing the role of enzyme and smartphone is the substrate. As we press, as we exert some pressure or as we apply some pressure on the smartphone, the balloon adjusts itself as per the size and shape, as per the shape of the smartphone. Here, balloon acts as active site or balloon play the role of an enzyme. Next one, substrate strain theory. This is similar with the induced fit theory. Substrate gets strained and results in cleavage of the bonds present in the substrate and thus increase the energy level of substrate. Here one, you have to concentrate that or you have to remember that in the same way as earlier stated in all the theories, enzyme get combined with substrate or enzyme acts on substrate, it forms ES complex. But here, after formation of ES complex, the substrate gets strained over the surface of enzyme, and due to such straining, there is cleavage of the bonds present in the structure of substrate, and such cleavage of the bonds present in the structure of substrate increases the energy level of the substrate as well as increases the mechanism of enzyme action. This is all about substrate strain theory. In mechanism of enzyme action, we have studied four different theories. Among those, first was enzyme substrate relationship theory. Second was lock and key model. Third was induced fit model. And next is substrate strain theory. If you have any questions or queries, you can contact to the given WhatsApp number or you can also write your queries through writing to this mail. Thank you very much.